Well, as you see, I am the only one speaking English today. But I have the pleasure to represent many of Yves Laporte's friends and collaborators from all over the world. From one of his best friends, Anders Lundberg from Sweden, to people from England like Peter Matthews or Baker, in the States, Ed Pearl, in Australia, Uwe Prosky, in Netherlands, Paul Verhoeven, and many others. When I first met Yves Laporte, that was in '61, and he and his collaborators, Paul Bessou and Bernard Pagès and François Semonet, were involved in one of the most remarkable series of experiments. These were experiments in which they compared responses of single sensory fibers innervating one particular muscle spindle in the muscle and trying to see the differences in the information that they forward to the spinal cord. And these experiments were really remarkable because they, not only because of the results they obtained, but also by the design of the experiments. They used a perfect preparation, and that was not so easy to find a muscle where the muscle spindles will be reasonably easy to analyze. We have heard many times today that Yves Laporte was perfectionist, that he wanted to have simple, be able to ask proper questions and to get quite unequivocal answers. So choosing the proper muscle where that doesn't work. Could you see? Well, in these particular muscles, I tried. Okay. Well, it works for you, but not for me. All right, but in these particular muscles, the muscle spindles are quite separated. They are arranged in a row. So one can try to find where the spindle is and try to dissect single sensory fiber that innervate single muscle spindle, dissect the dorsal roots in such a way that the single fibers can be recorded and be sure that they are coming from a proper muscle spindle. That was possible by combining sophisticated methods of recording from dorsal root fibers with a very simple technique of localizing them just by touching, by mechanical stimulation of the muscle to see from where the muscle spindle was activated. That was one of the procedures that were often used to combine what is the best from complicated techniques and from the very simple one, using whatever was available. So by using this technique, it was possible to record from, you can show the fibers at the top. One could record from single fibers that were analyzed, identified as primaries or secondaries, and see that they respond in different way. And then by using proper analysis, one could see the frequencies were changing, were different, that the sensitivity of the primaries was more phasic. They responded mainly when the muscle was stretched, and the secondaries were responding in more tonic way. So they clearly forward a different kind of information. Now, the question was following Yves Laporte's results, these results, which were quite extraordinary for me when I first heard about them. That was, in fact, a revelation that it is possible to find in the complex nervous system the way to analyze single fibers, single cells, networks of the single fibers and cells, and get something that one could, with confidence, tell about them. So we tried to find how the single fibers that were forwarding to the spinal cord this well-defined information, how these fibers influence cells in the spinal cord. But what we found was not so easy to make in agreement with the physiology of sensory fibers, because sensory fibers are designed and operate in such a way that provide a very quantified, very well segregated and defined information. But in the spinal cord, on neurons that we analyzed, we found that the information from such beautifully designed fibers is mixed. So that when we recorded from individual neurons, like the one, colored one, we found that the information from muscle spindle primaries, indicated 1A in green, and from group 1-2 afferents from the muscle spindles in blue, and also from tendon organs, is mixed. So that the neurons receive information from all three kinds of afferents and somehow integrate it. So now the problem was, if we look in the information to the whole population of these interneurons, we could see that in some neurons the information from the three kinds of afferents is mixed. They integrate information from all three. In some of them only from one, in some from two, in different combinations. So the question was to make sense of it. That was not particularly easy, and following Yves Laporte general ideas, we had to be sure that we look at proper neurons and properly identified neurons and not mix the information ourselves. Even if the information is mixed on the individual neurons, we should keep the neurons apart and be sure that we analyze them in separate neurons. And that was not so very easy because the motoneurons are easy to recognize in the spinal cord, but there are different population of spinal neurons, and it took us quite some time to find out which of them are doing what and to be able to recognize them. Right now we do not have quite complete picture of the spinal interneurons, but at least we know which of them are targets of primary afferents, secondary afferents, and the tendon organ afferents. So we can say that in most of these neurons, the information is mixed. Only in some of them, it is separated. But there are some ways to transform the neurons, integrating information from three kinds of afferents by reducing information from one of them by presynaptic inhibition or modular effects of monoamines. So the neurons can be transformed from integrating to more selectively processing the information. But nevertheless, the fact remains that the information is mixed. So we had to think how to explain the situation. And the explanation comes probably from the anatomy of sensory fibers, that once they enter the spinal cord, they branch. They give several axon collaterals. And these separate axon collaterals terminate on different kinds of neurons. So they have, one fiber may have something like 5, 6 or more kinds of target neurons, from motoneurons to 1A interneurons in green, to interneurons that process information from different kinds of neurons in violet, and 2 many other ones. And then there are subpopulations of excitatory, inhibitory interneurons, and so on. So the only explanation that we can give for the need for integration of information from the afferents is that different neurons need different combination of information to influence motor neurons or to influence neurons in the brain. And the information from individual kinds of sensory fibers must be integrated with different kinds of interneurons on this different kind of interneurons. And we hope that Monsieur Laporte would have been happy with this explanation. It is only one that we find now.